We are a team of researchers and students from UC San Diego. We're humbled by the fact that our school is the first in the nation to offer a specialized curriculum dedicated to nanoengineering. Our work focuses on exploring innovative themes in modern medicine, such as DNA nanotechnology, targeted drug delivery, and the deployment of viruses for cancer treatment. DNA Origami has been attracting much attention over the past few years. The origami structure consists of a long DNA strand, called a scaffold strand, and short DNA fragments, called staple strands. Complementary interactions of base pairs help the staple strands fold the scaffold strand into multidimensional shapes. DNA origami structures are biocompatible, stable, tough, and easy to program for different purposes, including targeted drug delivery, tissue engineering, and bioelectronics. Viral immunotherapy is one of the most widely investigated therapeutical platforms for cancer treatment. In 2015, the FDA approved the engineered virus T-VEC for the treatment of metastatic phase 3 melanomas. T-VEC is a type 1 herpes simplex virus engineered to selectively replicate in and induce lysis of tumor cells without causing damage to healthy cells. T-VEC is also able to promote systemic anti-tumor immunity by producing macrophage-stimulating factors around the lysed cancer cells. However, T-VEC immunotherapy is limited by its delivery system. In order to avoid instability in possible immune responses, T-VEC is delivered by intratumoral injection. This method poses a challenge for delivering T-VEC to metastatic tumors and organs that cannot be reached via injection. Inspired by this challenge, our team proposed the idea to use self-assembled DNA origami sheets and folic acid ligands as a multifunctional vector that can deliver the herpes simplex virus capsid and eventually the virus itself into cancer cells. Here's how it works. To form a single rectangular DNA origami sheet, 260 short DNA staple strands will bind to a long scaffold strand. Different sequences along the scaffold strand will pair with complementary staple strands, guiding the scaffold strand as it folds into the final rectangular sheet. To implement targeting abilities, 12 overhanging staple strands functionalized with thiol groups will be anchored to each DNA sheet. The thiol groups will then tightly bind to folic acid through effective click chemistry. With folic acid ligated on the origami sheets, our innovative DNA carriers can selectively target cancer cells. Next, our design takes advantage of the intermolecular forces between DNA origami sheets and the HSV capsid. The negatively charged DNA strands form a strong electrostatic attraction with the positively charged viral proteins that make up the capsid, leading to the tight packaging of the capsid within the origami sheets. With the viral capsid securely loaded, the DNA carriers are ready to move towards their target. As they come into contact with a tumor cell, the carriers will be engulfed via endocytosis. Inside the cell, the high pH environment causes the DNA complex to rupture, effectively releasing the viral capsid. If the real HSV virus is delivered using our DNA carrier system, the virus will be able to escape the endosome through its natural membrane fusion process. Once inside the cancer cell, the virus will replicate rapidly, causing the cell to burst.